Hello students, this is Ashita K. Sanuj, Assistant Professor, Department of Microbiology, EMA College of Arts and Science. This class we will be talking about tumor suppressor genes. Tumor suppressor genes are involved in many key cellular processes including regulation of transcription, DNA repair, cell-to-cell communication. The cell needs, if single defect is found in the cell can still control cell division. If both the genes of the cells are defected, only in that case the cell loses the ability to prevent division. All cancers demonstrate alteration in one or more tumor suppressor and oncogenes. So now let's get started with the example of tumor suppressor genes. The first example which we'll be talking is P50. P53, okay, let's write it down. 53. This was discovered in the year 1979. It is present in all type of cells. All type of cells. It is located or localized in the nucleus. Nucleus. It is located in the chromosome 17. Chromosome 17. Now, P53 produce protein product that function as transcription factor. The gene controlled by P53 is involved. The gene is involved in the cell division and viability. P53, this protein is going to interact with DNA and other protein, which will have this protein means which will have direct cellular actions, and if they find any DNA damage, they are going to trigger cell death or apoptosis. Well, it is found that P53 gene is found defective in about half of all tumors, regardless of their type or origin. That was actually found by many researchers, that the tumor, once it is found, and they have when they actually examined the tumor, they found out that P53 was found defective in all those tumors which they examined. Now, P53 protein, it is at the center of large network of proteins that actually sense the health of cell and cellular DNA. Now, what happens if they find there is a defect? Okay, that is a damage has been sensed. Is sensed. Now, the activity of P53 protein will aid in the decision between repair or induction of cell death. Okay, so it is actually this protein, it's actually stays along with a large network of proteins which actually sense the health of the cell or its cellular DNA. So if they find there is any defect in this particular cell or cellular DNA, what they're going to do, the P53, what it is going to do is that it will first, it will see whether it has to repair it, and if it thinks it cannot be repaired, it will actually allow the process of cell death. Okay, this P53 can stimulate transcription of group of target genes. Let me give you an example as P21. Now, actually, when it stimulates this particular gene, this will prevent the cell proliferation of P53. Okay, it is going to prevent. Okay, how it is going to prevent? The product of this P21 is a negative regulator of cyclin dependent kinase. Okay, this is very important. When it is going to stimulate P21, it is going to produce cyclin dependent kinase. This is an enzyme which is important for the cell cycle and cell division, right? So in case there is a damage, what will happen? That will prevent the cell proliferation by who? By P53. Okay. Now this P53, let me just put it on here so that I don't have to use another page. Okay, this P53, it can trigger the death in case of DNA damage. That is, if there is a damage, as mentioned, DNA is damaged. What it can do, it can directly take the cell to cell death. Okay, that is why it is called the guardian of genome. It's quite interesting to see that, right? Because if there is any damage, it has all the authority to take the cell to the programmed cell death phase. Okay, now if there is actually what P53 is removed or it is not there, what will happen? Then definitely if there is a damage, it will start accumulating the damaged DNA, right? The damaged DNA will be accumulated, and all the cells which are actually undergone division will all the cells will get the damaged DNA. Okay, so this P53, as it is a guardian of genome, it is very essential that it has to be in the active state. Now, how can you inactivate this particular gene? There are two ways by which you can inactivate. One is by mutation, another is by virus actually. Okay, now let's see how it does in the next slide. Okay, now let me just okay. So as mentioned, inactivation of P53. Inactivation can be done by mutation. Two ways in mutation itself. This is one day, okay? Mutation. And this, that if there is a mutation occurring in the promoter region and a mutation occurring in the protein coding region. Now, when it occurs, what a promoter actually, it controls how and when a gene has to be transcribed, right? So the mutation will actually decrease, okay, or there will be no or no P53, right? If it is actually mutation is occurring in the protein coding regions, it will have an impact on the expression of the gene, right? It is going to impact on the expression of the gene. And also it will decrease P53 as a transcription factor that we already know. And it will also affect, there is someone else who's going to get affected when the mutation is going to occur in this particular region. And that is P21. Also BAX. And next one is thrombospondin 1. This, it regulates the cell cycle and regulation. Let me write it, regulation. BAX, what it's going to do, it is going to induce or induction of apoptosis process. And this is an angiogenesis inhibitor. So when this activity, along with this, the activity of it also is lost, right? Not only that, it is going to affect these things. If you just consider the cell cycle regulation is lost, the induction of cell death also is lost, and also it is an angiogenesis inhibitor that is not going to inhibit angiogenesis, right? So all this will lead a cell to transfer into tumor or to cancer, you can say, right? Now, the other part by which this is one, okay, the other way when how you can actually inactivate P53 is the viral inactivation. How you can inactivate viral? By viral inactivation, how you can do? P53 is inactivated by several viruses. Most commonly mentioned is SV40. What is SV40? It is the simian virus 40, okay. And this virus is going to form or produce certain viral proteins, and they are actually called as large T antigen, okay. Now this will be produced in the cell cytoplasm. Now this will go and bind to, oh, why am I writing it like this? Okay. p53, and that will inactivate this gene. Okay. Apart from these simian viruses, hepatitis and human papilloma virus also does the same work of inactivating the p53. Okay. I hope now you're clear with p53. Now let's see the next um, tumor suppressor gene. The next one is APC, which is known as adenomatous polyposis coli. So it is actually a condition in which colon is filled with polyps. What are polyps? Are nothing but they are abnormal tissues, tissue growth actually, tissue growth on mucous membranes. In the case, it's in the colon membranes. So here this gene expression is very important for cell division. So if there is an inactivation of APC, so it is going to have over expression of the cell proliferation, which contribute to colon polyps. So actually, for a normal cell to become a cancerous, it needs many genetic alteration, several genetic alteration is required. So this APC protein, what it does, it will go and bind with beta catenin. Okay, now what is this beta catenin? Where this beta catenin is present in the nucleus. Okay, it has got a dual function, that is, it is involved in regulation plus in the coordination of cell to cell addition and gene transcripts. Okay, so what it does is this APC protein is going to bind with this B catenin. Okay, now it is a protein which is actually produced by one more thing I have to include here, by a gene called CTNNB1 gene. Okay, now what it does after that, it's going to form a complex, after which it forms, there will be a degradation occurring, degradation of this protein, that is beta catenin, will occur. Okay, that is in the presence of APC. But when it is not involved, okay, that APC is not present, what will happen? Definitely there will be a higher level of this beta catenin. In that case, this protein will go and bind to some other protein which is present in the nucleus. Okay, then it will go and bind and activate. It is going to activate the transcription factor, transcription factor of a gene which will control the cell growth and division. Okay. So what it does in the case of this, what happens, it is going to bind with this protein and forms a complex, and this protein will be removed out of the system. But once this APC protein are defective or it is not present, this protein level is going to shoot up. In that case, it needs to form a complex. So it will go and bind to some other protein in the nucleus to form a complex, and then it will bind to DNA and it will activate the transcription factor of the gene which controls the cell growth and division. Okay, you can just consider like, okay, there's a rough diagram. I can just, there's a cell, you have the nucleus. Inside the nucleus is what happens. Consider this as your beta catenin, and you have another thing which is a transcription factor 4. It's a protein coding gene. Okay, let's consider this. Okay, now what we have, if APZ is present, okay, in that case, what will happen? There will be no such, that is this is not free, right? So it will not produce any other protein, okay, so which will actually help in the proliferation of the cell. But once this APC is not present, what will happen? This binding to another factor will occur, and this will form another protein which is called the MYC protein. And you know about MYC, right? We already learned about it, which will help in the overexpression of the cell. Overexpression cells, so there will be more and more and more cells formed. Okay, now researchers have found that in the case of colon cancer, when they introduced normal APC proteins, there was a decrease in the tumor growth, tumor cell growth. Okay, so what the researchers did is like per individual who is suffering from colon cancer, they actually introduced normal APC proteins into their system, and which actually had a very good effect by reducing the tumor cell growth. And one more thing I would like to mention is this APC cell, APC genes. Okay, they such as they mediate the cell death of apoptosis. Okay, they, okay, apoptosis, that is, okay, let me just write it. Apart from this, apoptosis. Okay, so this is about the APC, which means the adenomatous polyps coli, which is associated with the colon field polyps, that is a colon cancer. And it expresses the uh, genes which are very essential for the cell division. When it gets inactivated, the colonic polyps, which are nothing but abnormal tissue growth on mucous membrane. Okay, so then I have told you about the all those things. So APC connecting with this, it gets degraded. If it is not, the increased level, this thing will be increasing. Then it will go and bind to another factor. Okay, and there will be cell growth and division, which is not good. And talking about treatment side, when you actually introduce the normal APC, it has shown to actually decrease the size or aggressiveness of the tumor. And I hope now you're clear with APC. Now let's go with another example. The next gene is the RP. This RP gene, that is a retinoblastoma gene. Then you should be familiar that is a cancer of eye, right? And it is mostly seen in young children. So this will actually encode a protein that acts by altering the activity of transcription factor. Okay, so it will interact indirectly in the control of gene expression. There are two forms of RB genes. Okay, the one is the sporadic, and the next one is the familial. Familial. Now let's see what are these. So sporadic type is that it can affect any individual, and it will depend upon the genetic changes and mutation. Okay, which an individual gets during its life span. Genetic change or mutation which an individual get during his lifespan. Okay, now the types of cancer you can see in this particular type is breast cancer, bladder, carcinomas. Here in this type, the mutations are comparatively relatively rare. A single tumor probability of getting is that a single tumor, okay, it will not have multiple tumors in this case, because here it is like in your cell, if you have genes coming from your father and mother, and it needs two mutations to occur, right? And that too in the same cell. So kind of probability is less that compared to if you have a defective gene already coming from your parents. So it just forms a single tumor here. In the case of familial, that is by the name itself, from family, it has been inherited. Okay, defective gene is inherited. In that case, it is inherited defective copy, defective copy of gene from parents, any parent, one of the parents. Now that you have already in your system that in a cell that you have a cell and this is okay and this has got some abnormality, okay, now it is passed on, so already there is one mutation occur, so you just need a one more mutation which has to occur in this particular gene, right? In that case, there will be probability that this individual can get many different types of cancer, especially osteosarcomas. In it, we will get mostly multiple tumors because I told you the reason here, right? And also, there will be recurrence of tumor. Okay, now retinoblastoma, RB, it stands for retinoblastoma. It's a gene which causes the cancer in the eyes, mostly seen in young children. And this is actually this codes encodes a protein which that actually acts by altering the activity of transcription factor, will interact with indirectly control of gene expression. There are few two types, okay, in the retinoblastoma, two different forms of retinoblastoma. That is, one is the sporadic and the one is the familial. Sporadic, sporadic is the one you should remember that the genes will be altered. It is not that you are getting it from your parents. Your cells are healthy ones, but during your lifespan, there are probability that one gene gets mutated, but still again the same cell, the other gene of the same cell has to be mutated in order to form any kind of cancer. Okay, but whereas in the case of familial, that is always inherited, that is already one gene is defective, though the chances that the other gene to get defective is high, right? So there will be multiple tumors and also recurrence of tumors. Now let me change the continuing RV genes. Normally, RV genes regulates the G1 cell cycle checkpoint. So actually it was seen in the case of breast cancer. Breast cancer, there was a deregulation in this checkpoint. Okay, that is, there was a deregulation seen in the G1 cell checkpoint, cell cycles checkpoint. Now, this RB gene product, which is known, which you can write it as PRB, normally it functions as growth inhibitor, functions as growth inhibitor by binding to transcription factor, binding to transcription factors. Okay, by binding to transcription factors. Now it is like it also have the activity to slow or halt cell division. It has got a major role in apoptosis. So this RB is like protein can regulate the transcription of RNA and transcription of rRNA and tRNA. That means they can control both control both, what is it, transcription plus post transcription events in cell. That is, they can regulate the transcription of regulate the transcription of rRNA plus tRNA. So they can control both transcription and post transcription events. And continuing, one more interesting thing is that it is associated with the chromatin modified protein, which is known as the HDAC. Okay, that means histone deacti deacetylase. Okay, so these RB genes are found in association with the chromatin modified proteins such as HDAC. Now, it will affect the, when it is, this association actually will affect the transcription by removing the acetyl group. Okay, the acetyl group will be removed. So it is going to affect the transcription, that is from histone. So it is going to affect the transcription. Now the cell closer association will be found between the DNA and the nucleosomes. Okay, that is, see this RB, it is going to have an association with the chromatin modified protein. Okay, chromatin modified protein, which is called a HDAC. And this is going to affect the transcription. It is going to affect the transcription by removing an acetyl group. Now the DNA and the nucleosomes will be much closer. Okay, now the closeness of this DNA and histone, the ratio of these two, or the interaction to be more specific, will actually, it is very difficult for the transcription factor. If because of this interaction, it is very difficult for transcription factor like E2F to bind to the target. In DNA, right? That is, it is going to make a defective when this ping E2F is not going to bind. It will definitely hinder other things, right? So that is how, that is what I mean to explain by all those things, right? So I think we'll move on to the last. Tumor suppressor gene, which is the BRCA. It gets its name from the association with the breast cancer. Breast cancer. So CABR, okay? There are two types in it, that is the BRCA1 and 2. So it is associated with breast cancer as well as with the ovarian cancer. Its main role is if DNA is damaged or the regulation of gene expression. So its main role is in the DNA damage or gene expression. Okay, so what it is going to do, it is if it these things, if the gene is non-functional, what will happen, as it is involved in this too, right? The DNA repair, the gene expression, just writing it like GE, gene expression will be compromised, right? Or will not be done. Now, in that case, what will happen? The DNA damage or will increase, and this will be passed on to the generation of cell, and it gets accumulated later, and it will happen in such a way that there will be a mutation occurring in the key genes, okay, which will lead to cancer. Hmm? So BRA, it is associated with the breast cancer, BR and CA. That's how it gets its name. There are two types, type 1 and type 2. It is also associated with the ovarian cancer, mainly if there is a damage, DNA damage, or if there is a faultiness in the gene expression, it is going to control it. So if a gene is non-functional, the DNA repair and the gene expression will not be occurring. As a result, the damaged DNA will be passed on to the generation of cells which gets accumulated, and as a result of which the mutation of few essential genes will occur, which will lead to cancer. And one more thing is that if cell lacks function functional functional BRCA gene, what will happen? It also causes the chromosomal breaks occur, or there will be too many centrosomes, which will actually interfere with the normal cell division and function. Okay, so one thing, if the damaged DNA, it is going to carry on with the future generation of the cells, and then cancer will occur. And if again it is non-functional, it can also cause the chromosomal breaks. Okay, it is going to cause the chromosomal breaks, and centrosomes will be piling up. That is, the level is going to increase. In that case, what will happen? The normal cell division and function will be interfered. And talking about the molecular structure, or sorry, the molecular level, these or you can say structure, there's no harm in it, they contain high proportion of repetitive repetitive DNA, which is quite uncommon or rare in human genes, and this will lead to genome instability and rearrangement. Okay, the more, so it will have many repetitive DNA, because of which it will not have a stable genome, and there will be a gene rearrangement. So the cancers which are associated with the defect of this gene are prostate, prostate, pancreatic cancer, cancer, well, colon. Okay. Now, another thing which I thought of mentioning here. Let me do it here. Yeah. So the association between this gene and the estrogen. Okay, what is the association? Is that uh, it plays an important role in the development of cancer in these tissues. Okay, so if during puberty and pregnancy in females, the estrogen level will be very high, which actually cause an increase in the breast epithelial cell proliferation. Okay, now which in turn places increased demand on the DNA repair capabilities of the cell and reproduction. Okay, now the reproduction of the cell with the lower DNA repair efficiently may lead to the formation of cancer. As I don't have space, so I'm just explaining it to you. I'll explain it once again. And this will, the association between the BRCA gene plus estrogen is very important in the development of cancer in these tissues. As the estrogen level are high during the puberty and pregnancy, that will actually cause an epithelial increase in the epithelial um, cell proliferation. Okay, proliferation, which in turn places increased demand on the DNA repair when there is a okay, so proliferation occurring, so it will have a negatively demand for the DNA repair capabilities of the cell. Now the reproduction of the cells with lowered DNA repair. Okay, so the reproduction occurs like some cells will have good DNA repair, or the damage has been done repaired, or the damage is more. Okay, so with lower DNA repair efficiency may lead to that will obviously form a defective cell, right? So it may lead to cancer. Okay, and see, as I mentioned you previously, it is like all the tumor suppressor genes. I think I mentioned it in my previous oncogenes video session, where I mentioned like here in oncogenes, like it in the sorry in the case of tumor suppressor genes, when you're getting a gene, okay, it depends upon the probability will depend upon how, like if you're getting it from your inheritance, okay, it is like inherited kind of defect you're getting it. That means one cell is already defective, and it just needs one more mutation for the other cell to turn into defectiveness. Okay, defective genes. In this case, the probability of getting cancer is high, right? So, but an individual who does not have any inheritance background, okay, the defective copy of the gene, all the cell carry defect genes. But what happens? It has to be like in the same phase. If you take it in the normal case, okay, the norm. I mean, this is normal, and this is the inherited inherited. It needs two mutations to occur, plus where it should occur in the same cell. Okay, so comparatively, there will be a probability of becoming this cell to tumor cell will be less compared to the one in this case, right? And now talking about the, I think I'll just wind it up with this treatment. It is the gamma radiation and drug called cisplatin and mitomycin all will interfere with the DNA damage of those drugs. Okay. This is the treatment. So with this, we have ended our video sessions on tumor genes. Okay, thank you.